Okay, children, good afternoon, everyone. Yesterday we have learned the aspects of pollination, right? Followed by different types of pollination depending upon the source of pollen, that is autogamy, okay, followed by heterogamy and xenogamy, right? And also we have seen how does the flowers get pollinated and what are the various agents of pollination. Okay, children. Now, when we look into the devices of what we call pollination, right, especially the autogamy, right. What is autogamy? Autogamy is the transfer of pollen grains from the anther to the stigma of the same flower, isn't it? So, now if this happens regularly, what will happen to the flower? That is, only the same characters will be transferred to the plant, isn't it? So therefore, continuous autogamy is not good for the plant. That is called self-pollination. Okay. So therefore, that will be always done with the help of its pollen will be falling on the stigma. Okay. So how can be there for autogamy? For example, there must be synchrony in the pollen release and the stigma receptivity, isn't it? Suppose if the autogamy has to take place, auto means self and gamy means union, the pollen release and the stigma receptivity must take place at the same time. And also then the stamen and that is the anther and the stigma must be close to each other. Now, already we have seen in some plants, like for example, that plants which have been what do you call having two different types of flowers. We can see the casmogamous flowers and clistogamous flowers, isn't it? Casmogamous flowers are those flowers which are having exposed anthers. No, I told you yesterday. Okay, isn't it? And clistogamous flowers are those which do not open at all. So therefore they have to show autogamy. So what is an advantage of this autogamy and what is a disadvantage of autogamy? Advantage is that there is assured seed set, isn't it? Even in the absence of pollinators. If there are no pollinators, even then what will happen? The pollination will occur because the flowers are closed. So the anthers of the same stamens, okay, will fall on the stigma of the same stigma, fall on the stigma. So therefore the self pollination may occur. Okay, but what is the disadvantage of that? The disadvantage is that we don't have any other characters. There will be no new characters formed in them. So these we can say as the devices for autogamy. Now what are the what do you call outbreeding devices? Let us see here. No. Now in some species, pollen release and stigma receptivity is not synchronized. Okay, that is a very important thing. Okay, pollen release and stigma receptivity is not synchronized. In the sense, both are not getting matured at the same time. The anthers will be getting matured earlier than the stigma, that is the gynecium, which we say the condition is called protandry. And we also have another condition called protogyny, where the gynecium gets matured from the earlier than the stamens. Okay, and moreover, in some species, the anther and stigma are placed at different positions. Okay, why they are placed in different positions? They are placed in different positions so that the anthers, the pollen grains of those anthers will not fall on the stigma of the same flower, and therefore preventing the self-pollination, preventing autogamy. So therefore, autogamy, having continuous autogamy causes inbreeding depression. That is a very important term. Okay, which we will learn in other chapters, later chapters. So that is called inbreeding depression. Okay, so therefore that is a very important aspect, that these two, that is the pollen release and stigma receptivity is not synchronized, and both the pollen, that is the stamen, and the stigma are placed in different what you call positions, so that the pollen cannot come in contact with the stigma of the same flower. Okay, so both these devices will be preventing autogamy. Okay, the third device to prevent inbreeding is self-incompatibility. What is this self-incompatibility? It is a genetic mechanism and prevents a self-pollen. That is a mechanism by which the pollen grains of the same flower may not be falling on the stigma of the same flower. Okay, usually when the pollen falls on the stigma has to respond to the pollen grains by secreting certain chemicals. There is a chemical interaction between the two. and that will be resulting in the growth of the pollen tube. So in that case, self-incompatibility is the what you call genetic mechanism in which self-pollination, okay, by preventing self-pollination by inhibiting pollen germination or pollen tube growth. Okay, and one more thing is that we can also prevent self-pollination by having production of unisexual flowers. No, what are unisexual flowers? Unisexual flowers are those where plants produce either the male flower or the female flower, right? Especially as in the case of cucumber, in case of papaya, etc., isn't it? And if both male and female flowers are present on the same plant, okay, which we call them as monoecious, okay, it can prevent autogamy but not geitonogamy, and in some several species, okay, which we say papaya, male and females present on different plants, okay, that is each plant is either male or female. What is this condition called? It is called dioecious, because the male plants are different, the separate, and the female plants are separate. So this can prevent both autogamy and geitonogamy. Okay, that means self-pollination and both the cross-pollination can be easily prevented by this condition. Okay, children. Now let us go into the other interesting aspect called pollen pistil interaction. Okay. Now we know that the during the what you call pollination brings about the pollen to the stigma of the what you call flower, isn't it? Okay. Now we know that when the stigma is providing the landing platform to the pollen, there may be number of pollens of the same species or different species falling on the stigma. So how does the stigma respond to the pollen grains of these plants? Okay, so when pollination does not, no, what is it? Pollination does not guarantee the transfer of right type of pollen. Okay, right. So therefore, the stigma has the ability to recognize the right pollen. So that has got the ability. The stigma has got the right ability to recognize the right type of pollen. That is what is called compatible pollen, which is suited for it. Okay, which is suitable for it. So the pistil accepts the right type of pollen and promotes post-pollination events leading to fertilization. Okay, pistil also can reject. Yes, pistil also can reject the wrong type of pollen, that is incompatible pollen, so that it cannot be able to grow. So how does the growth of the pollen tube or the compatibility of the pollen with the stigma is take place? It is occur by means of some chemical substances. Okay, both the pollen as well as the stigma, they respond to each other by certain chemicals, and these chemicals will be helping to respond to the changes that will be leading to formation of a pollen tube. Okay, children. Now let us see this figure 2.12. Okay, we can see the first picture here in A diagram. You can see a lot of pollen grains are falling on the stigma. Yeah, they are falling on the stigma, and then we can see long tubes coming out from that. It's called a pollen tube. Okay, so when pollen tube falls on the, what do you call, when pollen tube comes out from the pollen grain, so we say that the pollen germination has occurred, right? So what happens here? How does a pollen tube germinates? Let us see. Now look at this figure, that is 2.12 C diagram. Okay, so what is happening here? Here you can see the LS of ovule, the longitudinal section of the flower. Okay, showing the growth of the pollen tube. This is also longitudinal section of the female part, that is the showing the other growth of the pollen tube. Okay, she has shown the ovary, inside the ovule or inside there is a female gametophyte, that is embryo sac. Then it has got a style and a stigma on top. So what happens when the pollen grain falls on the stigma? Look at that. So many of these pollen grains, they will be producing the pollen tube. So how does the pollen tube comes out? Okay, and from where does it comes out? So we have learned that the pollen grain has got outer layer called exine and inner layer called intine. Okay, so the exine has got certain what you call germ pores where sporopollen is absent, isn't it? So through these germ pores, the pollen tube what you call generates. And at what stage these pollen grains are released actually from the anther? Okay, so usually we know that in many of the species they are shed at a 3-cell stage. Okay, what is a 3-cell stage? One, the large vegetative nucleus, which is also called tube nucleus, and the generative cell. The generative cell divides and forms what you call two male gametes. So as a result, what happens? these male gametes will be formed, and these two male gametes now are very essential for the purpose of fertilization. So the pollen tube, after falling on the stigma, it grows out, and then it passes through the style, and then slowly it enters into the ovule. When it enters into the ovule, it enters through the micropylar end of the ovule. Micropylar end of the ovule. Okay, and then enters in one of the synergids through which there is actually the filiform apparatus. Okay, and we know what is a filiform apparatus. Filiform apparatus are small structures that will be helping in guiding the pollen tube. You can see here. Okay, so filiform apparatus are there, so they will be guiding the male gametes to enter into the ovule, and then therefore entering into the female gametophyte, isn't it? So all these, isn't it? All these events from pollen germination on the stigma until the pollen tube enters the ovule, okay, are together referred to as a pollen pistil interaction. What is this called? Pollen pistil interaction. So what are the events? The events, right from the events where the pollen tube grows after falling on the stigma, passes through the style, enters into the ovule through the micropylar end with the help of filiform apparatus, and then releases the two male gametes into the embryo sac, and all these events are collectively called as a pollen pistil interaction. That is a very, very important aspect of the flowering plants. Okay, and how this interaction occurs? It is of course because of the compatibility of the pollen grain responding to the stigma, right? So when the compatible pollen grain falls on the stigma releases certain chemicals to recognize those what you call compatible pollen grains, and then allows the pollen grain to develop. Okay, so how this knowledge of pollen pistil interaction is very essential to a plant breeder. Okay, yeah, it is a very, very essential aspect to plant breeder. Why? Because it will be helping them to produce very good varieties of crop plants, right? So today in India and also in the world, we have got many different varieties of crops, isn't it? So how these different varieties of crops have been what you call done? How they have been raised? They have been raised by means of hybridization, which is known as artificial hybridization. So that is a very, very important aspect of what you call plants. That is the important aspect of the plant, isn't it? So what is this artificial hybridization? As a topic has come again, it is one of the major approaches of crop improvement program, isn't it? Where you have learned, you know about this word already before. It is nothing but the hybridization, where we cross some two different varieties of crop plants. As a result, they will be getting some new varieties, isn't it? So how it is done? Okay, now there are certain techniques. Okay, there are certain techniques by which we are able to do this, and since it is done by the plant breeders, by humans, therefore it is called as a artificial hybridization. Okay, so there are certain techniques to achieve this. It is called as emasculation and bagging techniques. Okay, what is this emasculation? Now see, if the female parent bears bisexual flowers, that is monoecious condition, both male and female are there. So what do? We will removing the anthers with the help of forceps. Okay, so by using a pair of forceps, we remove the anther before it dehisces. Okay, before it gets matured and before it get dehisces, we will be removing them. Okay, and this process of removing the anthers, okay, before they dehisces is known as a emasculation. E means removing, masculine in the sense male part, so therefore we are removing them. And after that, what do? We cover that flower with the help of a butter paper, and then okay, then that is done, that is called as a bagging. Okay, what is the advantage of this bagging? So when you cover the flower with the bagging, that is with the butter paper, you are preventing the stigma from unwanted pollen grains, isn't it? And you are preventing the contamination of the stigma with unwanted pollen grains. So since the air may bring different types of pollen grains onto the stigma, so you prevent it, isn't it? So this is how we can do this. And later on, what happens when the stigma gains what we call receptivity, desired pollen grains are brought, they have been removed, right? The bag is removed and it has been dusted, okay, onto the stigma, right? And then what do? We again, after dusting it, again we rebag. It is a very, very important thing. Okay, so after rebagging them, just wait for the process of fertilization to complete, and later on what you call the fruits will be formed. So that is a very, very important aspect of this. Okay, now if the female parent produces unisexual flowers, there's no need for emasculation, isn't it, children? Why? Because unisexual flowers means either they will be male flower or they will be female flower, isn't it? So this bagging or emasculation technique is done only in the case of bisexual flowers. Unisexual flowers, they no need to be done. Why? Because the flowers are separate and there is no need to, okay, done it. So done this. So when the stigma becomes receptive, pollination is carried out using the desired pollen and flowers are rebagged. Okay, children. Now let us see the next aspect called double fertilization. Okay. So why it is called double fertilization? No, it's very simple to understand because already in class 10th we have learnt about the double fertilization. Okay, now we know that the pollen tube, no, the pollen tube. Let us go back to the diagram once again. This one, yeah. Now look at that. This D diagram. Now in D diagram, okay, what is happening here? The male gametes are two male gametes are released into the embryo sac, isn't it? Now, what is the fate of these two male gametes into this embryo sac? Now, the first male gamete will fuse with the egg, okay, which is a part of the egg apparatus and forms the zygote. And this is known as syngamy. And since the first gamete is getting fused with the zygote, with the egg, therefore it is called as a first fertilization. Okay, and what about the other male gamete? The other male gamete will fuse with the polar nuclei. You can see here the two polar nuclei are present at the center, isn't it? So the second male gamete will fuse with the polar nuclei and forms what is known as a primary endosperm nucleus. It is called PEN. Okay, so since this actually involves three chromosomes, no, the fusion of three chromosomes, therefore it is triploid in nature. Therefore, how its triploid is written? It is written as 3N. So therefore 3N, okay, represents the triploid because it has got three sets of the chromosome. Okay, and therefore it is also called triple fusion. So therefore there are two fertilization in angiosperms, all flowering plants. One, the actual one, which where the zygote will be formed, where the zygote later develops into embryo, and then we have got the second fertilization, okay, where the second male gamete is fusing with the polar nuclei to form primary endosperm nucleus, which will be developing into endosperm later on stage to provide nourishment to the developing embryo. Okay, so two terms have to be remembered in this. Okay, first is double fertilization and triple fusion, which is the characteristic feature of all the angiospermic plants. Okay, children, later on the zygote develops into embryo, and later on the primary endosperm develops into endosperm, which provides a nourishment to the developing embryo. Right, children? So that's all for the today. Okay, so stay at home, stay safe and stay healthy. Thank you very much.